Hello everyone, welcome to Swati's Bioscience Hub. So in the upcoming series of videos, we are going to talk about cellular respiration. So cellular respiration idea is very simple, it's very basic. Okay. So the question is why we eat food. Okay, we eat food. So we need food because this food gives us energy. Okay, so when we eat food, this food get digested and it breaks down to three main macromolecules, which are carbohydrate, lipid and protein, with some a little bit of nucleic acid. So these macromolecules again metabolized and produced energy. Okay, this is the basic concept of cellular respirations. So these macromolecules like carbohydrates, lipids and proteins, they follow different pathways for the metabolism process to produce energy. Okay, and the cellular respiration process, it starts with glycolysis, then the conversion of pyruvate to acetyl coenzyme, then Krebs cycle, and lastly it ends with the electron transport chain. Okay, so today we are going to discuss the first step of cellular respiration, that is glycolysis. Okay, so when we see the word glycolysis, it comes from two Greek words. One is glykais and another one is lysis. Okay, one is glykais, another one is lysis. Okay, and this glykais means sugar or starch or sweet, and lysis means splitting. Okay, lysis means splitting. So basically sugar and starch or sweet, whatever we say, it's a form of carbohydrate. So it is a carbohydrate splitting process. Okay, and this glycolysis is basically, we understood from the word, that is a carbohydrate metabolism process. As I said, when food get digested, it breaks down to carbohydrate, lipid, protein mostly. So this is a carbohydrate metabolism pathway. So what happens? In this glycolysis, one glucose molecule is a 6-carbon compound. Okay, one glucose molecule, it converts to two pyruvate molecule, two molecules of pyruvate. Pyruvate is a 3-carbon compound. So here in this glycolysis process, one 6-carbon compound glucose converts to two number of 3-carbon compound pyruvate in a series of enzymatic reaction. Okay, so this is the process of glycolysis. This glycolysis mostly occurs in the cytosol of the cell. It takes place in the cytosol of the cell and also known as Embden-Meyerhof pathway. Glycolysis also known as Embden-Meyerhof pathway. Meyerhof pathway. Okay. So what we get in the glycolysis is a type of carbohydrate metabolism, and also it is an oxygen independent reaction. Why? This reaction does not involve any molecular oxygen. Okay, it does not involve molecular oxygen, so it can take place in aerobic condition as well as in anaerobic condition. Okay, it is an oxygen independent reaction. So it goes through a series of enzymatic reactions. Okay, clear? So this is the basics of glycolysis. So now we'll discuss all the enzymatic reaction steps which are involved in the conversion of glucose to pyruvate. So let's start with the steps in glycolysis. So as I said, it takes place in cytosol of the cell, and glycolysis mostly it's the process of conversion or splitting of sugar splitting of one glucose molecule to two pyruvate molecule. Okay, so first one glucose molecule, glucose is a 6 carbon compound molecule. Okay, so one glucose molecule will convert to glucose 6-phosphate. It will convert to glucose 6-phosphate. That means here this glucose molecule accept one phosphate group here. That phosphate group added to the 6th carbon position and it becomes glucose 6-phosphate. So here it accept one phosphate group from ATP and in the process of phosphorylation. Okay, and the enzyme responsible here is, I am writing the number of enzyme here and writing the name of the enzyme here. Okay, the enzyme responsible here is hexokinase or glucokinase. Hexokinase or glucokinase. Okay, and this hexokinase or glucokinase, it requires magnesium ion or manganese ion for its activation. Okay, these ions activates these enzymes, then this enzyme stabilizes this reaction from glucose to glucose 6-phosphate. We write this towards this side a little bit. Just a bit. One glucose molecule, it accepts a phosphate group from ATP and it produces glucose 6-phosphate. Okay, the presence of enzyme 1. Then this glucose 6-phosphate will convert to one of its isomer. Okay, this glucose 6-phosphate will convert to one of its isomer because the carbonyl oxygen which is present on the first position of first carbon of the glucose, the carbonyl oxygen which is present on the first carbon group of the glucose, it shifts to the second carbon group. So this glucose becomes fructose. This glucose becomes fructose and it becomes fructose 6-phosphate. Okay, it's a isomerization process. As these are the isomers of each other, this is a reversible reaction. And the enzyme which is responsible for this reaction is glucose 6-isomerase. Just a minute. Phosphoglucoisomerase. Okay, the enzyme responsible here is phosphoglucoisomerase. It converts from glucose 6-phosphate to one of its isomer, that is fructose 6-phosphate. Now this fructose 6-phosphate will accept one phosphate group again from ATP. Okay, it will accept one phosphate group and here it will become fructose 1,6-bisphosphate. Okay, it becomes fructose 1,6-bisphosphate. Okay, because there are two phosphate groups are present, and this one phosphate group is added at the first carbon position of the fructose 6-phosphate, and this takes place in a phosphorylation process. Okay, the conversion is a phosphorylation process, and the enzyme responsible here is phosphofructokinase. The enzyme responsible here is phosphofructokinase. As we know that kinase is responsible for phosphorylation process. Okay, the enzyme is phosphofructokinase. Now here we have the 6 carbon compound. At this stage, the sugar splits or clipped into 3 carbon compounds. Okay, it splits to 3 carbon compounds. One is glyceraldehyde 3-phosphate and another one is dihydroxyacetone phosphate. Okay, one molecule of glyceraldehyde 3-phosphate and one molecule of dihydroxyacetone phosphate, and these two molecules are 3 carbon compound. Okay, 6 carbon compound get clipped or splitted to produce 2 molecules. One is glyceraldehyde 3-phosphate, another one is dihydroxyacetone phosphate. And the enzyme responsible here is aldolase. Okay, the enzyme responsible for the splitting is aldolase. Okay, now the thing is, this dihydroxyacetone phosphate is an isomer of glyceraldehyde 3-phosphate. So here what will happen, this dihydroxyacetone phosphate, there is an enzyme which converts this dihydroxyacetone phosphate to its isomer glyceraldehyde 3-phosphate. Okay, and the enzyme is known as triose phosphate isomerase. Okay, triose phosphate isomerase, because here isomerization process takes place. Okay, it does isomerization from DHAP dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Now these two molecules of glyceraldehyde 3-phosphate, what will this do? 2 molecules of glyceraldehyde 3-phosphate. Okay, these 2 molecules of glyceraldehyde 3-phosphate will convert here it will accept one phosphate group. Okay, but here there is some changes. Here it will not accept that phosphate group from ATP. It will accept the phosphate group from monohydrogen phosphate. Okay, here it will accept the phosphate group from 2NAD plus to HPO4 2 minus. That means monohydrogen phosphate. So here from this monohydrogen phosphate it will accept that phosphate group, and here this NAD plus will convert to NADH, and after the phosphate group removal from monohydrogen phosphate it will release hydrogen ion, and then this glyceraldehyde 3-phosphate will convert to 1,3-bis phosphoglycerate. It will convert to 1,3-bis phosphoglycerate. Okay, this two molecule of glyceraldehyde 3-phosphate will accept one phosphate group from monohydrogen phosphate, and from this it will convert to 1,3-bis phosphoglycerate. Means there is, at the third point there is this phosphate group was present, but now at the first carbon also this phosphate group is present, and it is a reversible reaction. Now this 1,3-bis phosphoglycerate. Now this 1,3-bis phosphoglycerate will release one phosphate group from itself. It will release one phosphate group from itself. That means from ADP, from ADP here, ADP converts to ATP in the form of energy. So here it will remove one phosphate group, ATP accept that phosphate group, and it converts to ATP, the form of energy, and it converts to 2 number of 3-phosphoglycerate. Okay, it converts to 2 number of 3-phosphoglycerate. Now the enzyme which is responsible for the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate is known as, it is a fifth number, sorry, it is a fifth number enzyme. So here this is a 6th number enzyme. The enzyme which is responsible for conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate is glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. Okay, and the enzyme which is responsible for this conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, that enzyme is known as phosphoglycerate kinase. Phosphoglycerate kinase. Here kinase enzyme acts, that means it is a phosphorylation process. Removal or acceptance of phosphate group is a phosphorylation process, and this is a substrate level phosphorylation. This is a substrate level phosphorylation process, and here it removes one phosphate group in the form of ATP to produce ATP, and this 1,3-bisphosphoglycerate will be converted to 3-phosphoglycerate. Now this 3-phosphoglycerate will convert to one of its isomer. Okay, so the what will happen, the phosphate group which is present at the 3rd carbon, it will convert to a mutant group. Okay, 3-phosphoglycerate will convert to one of its another molecule, that is 2-phosphoglycerate in the process of mutation. Okay, it will convert to 2-phosphoglycerate in the process of mutation, and here the enzyme responsible for this mutation process is known as phosphoglycerate mutage. Phosphoglycerate mutage. So the phosphate group which was present at the 3rd carbon position, here this phosphate group will be shifted to the 2nd carbon position. So it is a mutation process, and in this process this 3-phosphoglycerate will convert to 2-phosphoglycerate in the presence of phosphoglycerate mutase. Okay, now this 2-phosphoglycerate will convert at this process, this 2-phosphoglycerate, it will remove another phosphate group from itself, and the ADP will accept that phosphate group and produce ATP. Okay, and the 2-phosphoglycerate will convert to phosphoenol pyruvate. Phosphoenol pyruvate. Okay, it will convert to phosphoenol pyruvate, and this is also a substrate level phosphorylation process. Okay, and the enzyme responsible for here is enolase. The enzyme responsible for conversion of 2-phosphoglycerate to phosphoenol pyruvate is a enolase. Now this phosphoenol pyruvate, and this phosphoenol pyruvate, the phosphate group is there. Okay, so here what will happen is 2-phosphoglycerate will convert to phosphoenol pyruvate, but here it will remove one water molecule from itself. I'm really sorry for that. So for the inconvenience, the phosphoglycerate will convert to phosphoenol pyruvate because it release one water molecule here. Now this phosphoenol pyruvate will release one phosphate group from itself. Okay, at this position the substrate level phosphorylation takes place, and this phosphoenol pyruvate, it will release one phosphate group from itself, and ADP accept it and produce ATP, and this phosphoenol pyruvate will convert it to pyruvate, the final product of glycolysis process. Okay, is responsible for this conversion of PEP or phosphoenol pyruvate to pyruvate is known as pyruvate kinase. It's a substrate level phosphorylation and it is known as pyruvate kinase. And this pyruvate kinase, it requires magnesium or manganese ion. It requires magnesium or manganese ion and potassium ion for the activation process. Okay, for this activation, this pyruvate kinase requires magnesium or manganese and potassium. Okay, potassium must be there and either of this manganese or magnesium. So here this pyruvate kinase will remove the phosphate group from the phosphoenol pyruvate, and the phosphate group will be accepted by this ADP and produce ATP, and then this phosphoenol pyruvate produce the pyruvate, the final product of glycolysis. So now from one glucose molecule we get pyruvate. Okay, this is the glycolysis process. So let's summarize once again. One glucose molecule, it will undergo phosphorylation process, and here it will produce glucose 6-phosphate, and at this process it removes one phosphate group from itself, and the enzyme which is responsible for this conversion is hexokinase, specifically glucokinase, that requires magnesium or manganese ion for the activation. Now this glucose 6-phosphate will convert to one of its isomer, that is fructose 6-phosphate, and the enzyme responsible for it is a phosphoglucoisomerase. Okay, and this fructose 6-phosphate again undergoes phosphorylation process. Here it accepts one phosphate group from ATP and converts to fructose 1,6-bisphosphate, and the enzyme responsible is phosphofructokinase. Now this fructose 1,6-bisphosphate will get splitted. Okay, this sugar gets splitted to produce one molecule of glyceraldehyde 3-phosphate and one molecule of dihydroxyacetone phosphate. Now this dihydroxyacetone phosphate will again convert to one of its isomer, that is glyceraldehyde 3-phosphate. And the enzyme responsible here is triose phosphate isomerase, and the enzyme responsible for splitting process or the cleavage process is known as aldolase. Okay, and at this point this 2-glyceraldehyde 3-phosphate will convert. At this point it will accept one phosphate group, but not from ATP. It will accept that phosphate group from monohydrogen phosphate, and here it converts to 1,3-bisphosphoglycerate. Okay, and the NAD converts to NADH and it releases one hydrogen ion. And the enzyme which is responsible here is glyceraldehyde 3-phosphate dehydrogenase. Now this 1,3-bisphosphoglycerate will undergo substrate level phosphorylation. At this point it removes one phosphate group. ADP accepts that phosphate group and converts to ATP. This 1,3-bisphosphoglycerate removes the phosphate group from the first carbon. It becomes 3-phosphoglycerate. And the enzyme which is responsible here is phosphoglycerate kinase. Okay, and here this phosphoglycerate will again mute. It undergoes mutation process to produce 2-phosphoglycerate. The phosphate group at the third carbon position shift to the second carbon position, so it becomes a 2-phosphoglycerate, and the enzyme responsible here is phosphoglycerate mutase. Now this 2-phosphoglycerate will remove the water molecule from itself in the presence of allolase, and it converts to phosphoenol pyruvate. At this point, this phosphoenol pyruvate will again remove that phosphate group from itself. Okay, and the ADP accepts that phosphate group, release ATP in the form of energy, and the phosphoenol pyruvate converts to pyruvate. So now we can see that one glucose molecule produces 2 pyruvate molecules. Okay, so this is the stage of glycolysis. Now we'll analyze the reaction of the net reaction of glycolysis. If we will see the net reaction of glycolysis, we can say one glucose molecule. Okay, we'll first remove it. First remove it, then we'll discuss the net reaction, where one glucose molecule is cleaved or lysed to produce 2 pyruvate molecules. Glucose molecule is a 6 carbon compound and pyruvate is a 3 carbon compound. So the net reaction of glycolysis is 1 glucose molecule plus 2 ADP plus 2 NAD plus, okay, plus 2 HPO4 2 minus, that is monohydrogen phosphate. It produces 2 pyruvate, 2 ATP, 2 NADH plus hydrogen ion. Okay, so in glycolysis we are getting 2 ATP and 2 NADH. ATP is the form of energy. So here in presence of aerobic condition, that means in the presence of oxygen. Okay, oxygen molecule, when oxygen molecule is present, at that point NADH will be oxidized to produce ATP. So in presence of oxygen, 1 NADH get oxidized to produce 2.5 ATP. Okay, so in aerobic condition we are getting 2 ATPs directly from 1 glucose molecule. We are getting 2 ATPs directly plus 2 NADH, that means 2 into 2.5 ATP, that means 2 ATP plus 2.5 ATP, that is 5 ATP. We are getting total 7 ATP in aerobic condition. From 1 glucose molecule we are getting 7 ATPs from in aerobic condition. But in anaerobic condition, that means when oxygen molecule is not present, at that condition we'll get from 1 glucose molecule we'll get only 2 ATP because NADH cannot be oxidized here. So we'll get that 2 ATP that is directly produced from the glycolysis process. So this is the a basic idea, the steps of the glycolysis process. So today in the video we have discussed the steps of glycolysis process, the basic of glycolysis process, the enzymes which are responsible for these reactions involved in the glycolysis process, and the net reaction of glycolysis, and at last we have discussed the ATP which we are getting from the glycolysis process both in aerobic and anaerobic conditions. So this is it for today's discussion. If you have any doubt regarding today's discussion, please comment in the comment box. Please like and share the video and don't forget to subscribe my channel Swati's Bioscience. Thank you so much.